Hey there, biology enthusiasts. Today, we are diving into one of the most fascinating molecules in biology, DNA. Ever wondered how your body carries all the instructions for everything you are? From your eye color to your ability to digest food, DNA is the code behind it all. DNA, or deoxyribonucleic acid, is like the instruction manual for life. It's the molecular blueprint that determines our traits and controls our growth. Imagine a library filled with millions of books that describe how to build a living organism. That's DNA. And just like a recipe for your favorite dish, DNA holds the exact steps to build proteins, the building blocks of life. It's amazing that all of this information is wrapped up in a tiny molecule inside our cells. The human genome contains over 3 billion base pairs of DNA. Yes, you heard that right, 3 billion base pairs. If you stretched them out, they would be over 2 meters long. How does such a long molecule fit inside such a small space? Let's explore. Now, let's zoom in and break down the chemical structure of DNA. Picture a nucleotide as a small building block of DNA. Each nucleotide has 3 parts: a phosphate group, a sugar molecule, and a nitrogenous base. There are 4 types of nitrogenous bases: adenine, A, thymine, T, guanine, G, and cytosine, C. These nucleotides join together to form a polynucleotide chain linked by a phosphodiester bond between the sugar of one nucleotide and the phosphate group of the next. The bases are the key players: adenine pairs with thymine and guanine pairs with cytosine. Why is this important? It's because these base pairs are the key to storing genetic information. AT and GC form the steps of the twisted ladder we call the DNA double helix. Imagine this process like a zipper where each base pairs up with its partner, ensuring everything is in the right place. Let's talk about the real showstopper, the double helix. In 1953, scientists James Watson and Francis Crick proposed this iconic structure of DNA. Picture a spiral staircase. Two long chains of nucleotides twist around each other to form a helical shape. This twist is what gives DNA its strength and stability. The cool part? The 2 strands of DNA run in opposite directions, one from 5' to 3' and the other from 3' to 5'. This is called antiparallel orientation. The bases in the middle are held together by hydrogen bonds, creating base pairs like A with T and G with C. Just imagine, this double helix structure is so strong, yet so flexible, enabling DNA to carry all the genetic instructions for life. It's almost like a tightly wound rope that can store vast amounts of information while still being able to unzip when it needs to replicate or make proteins. Here's a fascinating aspect of DNA, its antiparallel nature. This means that the 2 strands of DNA run in opposite directions. One strand has a 5' to 3' direction, and the other runs 3' to 5'. Why does this matter? This orientation is critical for DNA replication. The DNA strand acts as a template to create a new strand, and the antiparallel direction ensures that enzymes can copy the genetic code in the correct direction. Without this, the cell wouldn't be able to make exact copies of DNA during cell division. Imagine a one-way street. Just like traffic can only flow in one direction, the DNA polymerase enzyme moves in one direction along the strand to ensure accurate copying. Now, let's step back and appreciate how DNA is packed inside the nucleus of eukaryotic cells. DNA is long, really long. If you stretched out the DNA in one human cell, it would be about 2 meters. But how does it fit inside a tiny nucleus? Here's the magic. DNA is wrapped around special proteins called histones, forming structures called nucleosomes. These nucleosomes look like beads on a string under a microscope, and they help package the DNA into a compact form called chromatin. As the cell gets ready to divide, the chromatin condenses further into visible chromosomes, making it easier to replicate the DNA and distribute it equally between new cells. It's like organizing a tangled ball of yarn into neat coils. This packaging ensures the DNA fits neatly into the nucleus while remaining accessible for vital processes. Now that we've seen how DNA is structured and packaged, let's dive into its role in the central dogma of molecular biology. The central dogma states that genetic information flows from DNA to RNA to protein. First, DNA is transcribed into messenger RNA, mRNA, in the nucleus. Then, the mRNA travels to the ribosome, where it's translated into a protein. Proteins are the workhorses of the cell, carrying out essential functions like catalyzing reactions, building structures, and even defending against diseases. Imagine DNA as a recipe book, mRNA as the chef's instructions, and the protein as the final dish. This flow of information from DNA to protein is what makes all the processes of life possible, from your muscles contracting when you move to your body fighting off infections. So, there you have it. From the double helix structure to how DNA is packaged inside the cell, to the central dogma explaining how genetic information gets translated into action, DNA is truly the foundation of life. Isn't it incredible to think that this tiny molecule holds the instructions for every trait, function, and process in our body? Now you can appreciate how DNA replication allows for accurate copying of genetic material and how proteins are the end product of this amazing process. DNA doesn't just carry genetic information, it brings life to every single cell, ensuring that life continues and evolves. So, next time you hear about DNA, remember, it's not just a molecule. It's the blueprint for life, driving everything from your hair color to how your cells respond to the environment. Until next time, keep marveling at the wonders of biology.